fakultas terbaik di nasional dan juga dunia ya. Dan ini juga kemarin saya sempat bersilaturahmi juga dengan Pak Dekan. Nah ini sangat bangga, banyak program-program baru yang selalu cepat diadop oleh FK USK ya, dan termasuk pemeriksaan-pemeriksaan update yang ada di Prodia itu juga didukung terutama dari riset-riset yang ada di Fakultas Kedokteran USK. Sebenarnya hal ini juga sangat sejalan dengan misi Prodia untuk diagnosa lebih baik dan untuk Sidia yang bergabung dengan Prodia dan visi Prodia sebagai Center of Excellence. Dan Prodia semangat menuju perubahan next generation healthcare yang handal dengan 7 filosofi kita yang sangat luar biasa, dan yang paling utama adalah mempertahankan kualitas sebagai budaya kerja dan orientasi pada kebutuhan pelanggan. Dan Prodia dalam hal ini selalu siap dokter menjadi partner untuk USK khususnya. Dan saat ini sudah lebih dari 60.000 ya benar dokter senasional yang telah bekerja sama dengan Prodia, dan tentunya kerjasama ini jugalah yang mendorong banyaknya kerjasama dan juga kerjasama riset tentunya dan juga publikasi. Dan kita sudah mencatat kerjasama dengan 43 institusi kedokteran dan termasuk 15 tahun dengan FK USK, dan ini suatu prestasi yang sangat luar biasa. Baik juga dengan komunitas-komunitas ilmiah yang berada di bawah naungan FK USK itu sendiri. Dan tentunya hal ini sekali lagi sangat mempercepat pemeriksaan tes-tes baru yang ada di Indonesia. Akhir kata sekali lagi saya mewakili Prodia Sumatera mengucapkan selamat atas terlaksananya webinar untuk tahun 2023 ini sebagai kuliah tamu kami untuk Fakultas Kedokteran USK. Sekali lagi terima kasih juga atas waktu yang telah diberikan. Nah, waktu dan tempat saya kembalikan ke Bapak MC, Dr. Musin. Silakan dok, terima kasih.
Baik, Ibu Marisa, terima kasih banyak atas sambutannya. Mudah-mudahan seperti saya sampaikan, kerjasama dengan FK USK akan semakin berkembang nantinya. Kita ke kegiatan utama kita pada hari ini, yaitu presentasi oleh Profesor Gerard Pals, PhD, yang bertajuk From Research to Publication. Sebelumnya saya akan membacakan CV daripada moderator, karena pada kesempatan kali ini kita akan diskusi kita akan dimoderatori oleh Dr. Nanda Ayu Puspita, MKes PhD. Beliau adalah staf di bagian biokimia Fakultas Kedokteran Universitas Kuala. Saya bacakan CV beliau. Beliau menamatkan sarjana kedokteran dan dokter di Universitas Padjadjaran Bandung pada tahun 2005. Kemudian melanjutkan S2 Biokimia Medis di universitas yang sama di Bandung dan menyelesaikan program doktor atau S3 di University of Salford di Manchester, Inggris, dan saat ini sedang dalam program postdoktoral di University of Teesside di Middlesbrough di UK di Inggris pada topik proteomics scientist pada topik proteomics dan beliau sebagai scientist di bidang proteomics. Dan pekerjaan beliau adalah sejak tahun 2005 sampai sekarang beliau adalah dosen di biokimia di Fakultas Kedokteran Universitas Kuala, dan kolaborasi proyek industri dalam 5 tahun terakhir yang terbaru adalah dalam proyek proteomics analysis of F. venenatum dengan partner adalah Teesside University UK dan Marlow Foods. Kemudian dengan publikasi beliau dalam 5 tahun terakhir, yang paling terbaru adalah jurnal dengan judul Differential Proteomics of Helicobacter pylori Isolate from Gastritis Ulcer and Cancer Patient, First Study from Northwest Pakistan, yang dipublish di jurnal Medicina tahun 2022. Beliau juga sudah menerbitkan banyak publikasi internasional seperti yang ditampilkan dalam slide ini. Untuk menghemat waktu, saya persilakan kepada Dr. Nanda Ayu Puspita, MKes PhD, untuk memimpin jalannya diskusi kita pada hari ini. Saya persilakan kepada dokter.
Baik, terima kasih Dr. Muhsin atas kesempatannya. Assalamualaikum warahmatullahi wabarakatuh. Pertama-tama saya mengucapkan terima kasih atas kesempatan yang diberikan kepada saya dan terima kasih sebesar-besarnya juga kepada Prodia yang sudah menyelenggarakan webinar ini. Memberikan kesempatan kepada kami para tim akademisi di Universitas Syah Kuala untuk ikut berpartisipasi dalam program besar Prodia untuk research ini. First of all, I would like to thank Prof. Gerald that has provided and his valuable time to be with us here. This is still morning in Netherlands, so we really appreciate your time to join us this early morning. And I know this isn't an easy time in Netherlands at this time because it's still winter, so it's still kind of dark outside. But yeah, hopefully this room will make us all like warm, and we're all ready to learn from you. Sebelumnya saya akan membacakan juga CV dari Profesor Gerald. Ya, Profesor Gerald merupakan peneliti besar yang sudah mempunyai karir panjang di dunia penelitian, baik dalam bidang industri ataupun di bidang akademisi. Beliau menyelesaikan pendidikan S1 di biologi dan kimia pada tahun 1977. Kemudian melanjutkan program S2 biokimia di Universitas Utrecht, Belanda, dan menyelesaikan S3 di bidang genetika manusia di Amsterdam juga. Saya tidak bisa membaca universitasnya. Prof. Gerald, I'm trying to pronounce your university when you finish your PhD, but I'm afraid I'm not going to do justice to read it.
It's Vrije Universiteit.
Vrije Universiteit, all right. Oke. Selanjutnya untuk riwayat pekerjaan, beliau juga sudah mempunyai riwayat karir yang sangat panjang di bidang research. Pada tahun 2007 sampai 2016, beliau merupakan kepala penelitian untuk penelitian connective tissue research. Kemudian dari tahun 2016 sampai 2020 beliau juga aktif bekerja sebagai konsultan di bidang penelitian untuk genetik dan juga termasuk ikut berpartisipasi dalam penelitian COVID testing di Netherlands. Kalau saya bacakan sepenuh-penuhnya penelitian beliau ini sangat panjang sekali, tapi saat ini beliau menjadi salah satu kurator di International Database of DNA Variant dalam bidang osteogenesis imperfecta. Dan beliau juga menjadi visiting professor di 2 universitas di Indonesia. Publikasinya sudah bisa kita lihat sangat banyak, lebih dari 300 publikasi yang dikutip lebih dari 20 kali dengan H-index-nya 64. Dan salah satu penghargaan yang beliau terima adalah pada tahun 1995, yaitu beliau mendapatkan penghargaan sebagai ahli genetika molekular klinis. Baik, untuk hari ini Prof. Gerald will guide us through about research to publication, and it is an honor for us today to be listening to your lecture today. That will be, I believe, the time allocated for you is around 40 minutes. Yeah, but I think we don't mind if we go a little bit beyond that 40 minutes. All right. So without further ado, I would like to welcome you to give your presentation, Prof. Gerald.
Thank you. I will share my screen. The right one. Can you see my screen now?
Yes, we can
Yes,
see
Prof.
your screen.
Okay. So I will start with some very basic things. And my presentation is based on the fact that in order to be able to publish your research, you need to think already about publication before you start research. And that's a way of thinking that many people do not have. People jump into the research project, and when they have collected the data, they suddenly realize that it may not be publishable. So I have to find the right button now to go to the next slide. Oh, yeah, okay. So the design of research aimed at publication. I give some examples of research and publication. I will talk about how to write an English manuscript with some very basic errors that I often see in people who try to translate the text from Bahasa Indonesia. And very important, how to find the right journal. So why do we perform research? To collect or generate data. It can be important for patient care, of course. To increase our knowledge, important point, of course. If we have grants for research, it's required to publish the results. And curiosity. And why do we publish research? To share our data. It's also important for patient care, of course. To share our knowledge. It's also required for grants. And in the university world, we call this publish or perish. To keep your job, if you have a job at university, you want to keep it in. So before jumping into a research project, sorry, what is it I want to know? Is the answer relevant to others? Is it possible and likely to get it published? And very important issue, what is the current state of knowledge? If you forget about that, you get results that have been published by someone else already, and you don't succeed in getting it published. So it's also important to see what makes us special in the research field. What is it I want to know? If you are not curious about the outcome of your research, it gets boring. Is the answer relevant to others? So think about it. What will the possible outcomes of your research be, and what is the impact of the possible outcomes of the patients, the scientific community, your grant application? Is it possible and likely to get it published? Well, think of the possible outcomes. If you were editor or reviewer, would you consider a paper on this subject news or even big news? And then to find out what is the current state of knowledge, you need to read. So check the recent literature. Is my idea new? Will the outcome add to the current knowledge? And if publications are not accessible for you, you can ask the authors. There's always an email address in the publication, or you can ask me sometimes for a PDF by email. We used to do that with little reprint cards that we sent to the author to ask for a reprint. And when we published something in the old time before internet, we had to buy for very expensive reprints of the publications. And we had little cards that had pre-printed our address. And I kept some of those because from the East Bloc, in the time that there was still what we call the Iron Curtain across Europe, the countries, the scientists from the Russian area were not allowed to write in English. So they would send us little cards in Russian with a translation, not in English, but a translation in Latin, which was the old-fashioned language of science from the 17th century. What makes us special? Very important. Do we have, for example, if you do medical research, unique patients? Yes, we do. Do we have unique facilities? Well, we have very nice facilities, but if you look at the rest of the world, it's not really unique. Do we have unique populations? Yes, we have a lot of unique populations, 354 recognized ethnicities in Indonesia. Do we have unique research questions? Well, of course we do, but you have to work on that. Do we have a unique biobank? That also needs work, but there's a good start at PRODIA, I think, so that's also important. I will give you one example of a special population in the Netherlands, because the Netherlands is a tiny country, but we still, inside the country, we have some places that are genetically isolated. This is a very old picture, of course, this Volendam. And this village has been isolated by religion. When in the 16th century there was a reformation in the church and the Netherlands became an independent country, this village was isolated and remained Roman Catholic. And they have a problem that there is it's called a founder effect. 1 in 400 people there is affected with what we call primary ciliary dyskinesia, a very rare disease. It's very common in this village, but other diseases, other inherited diseases, are not found there at all. This is the primary ciliary dyskinesia. I won't go into that, but the fact it's a very complicated disease. The cilia have hundreds of proteins, so there are hundreds of genes involved. But we found a new gene in this population, in the CCDC114 gene. And I'm showing you this because this paper that we published in 2013 has the conclusion in the title. So it's something to think about when you write a publication. If you have an interesting conclusion, you may already put the conclusion in the title and draw attention to it. It's in a genetics journal, that's also important. This was a new gene and it was important in the field of genetics. If this gene had already been known in the field of genetics, we might have chosen a different journal, a journal in pulmonology, because it's important for the lungs. And this one had a very high impact factor. I know that in Indonesia, people often look at the quartiles, the Q1, 2, 3, and 4. I think that's from Scopus. I never heard of it before. I worked in Indonesia, but in Europe and the United States, we are looking more at what we call the impact factor. So the impact factor is the average number of citations that you get from a publication in that journal, I think in the first year after publication. An example of research, Marfan syndrome is a disease where people look a bit different, very often, not always, by the way. They have long limbs, long fingers, but the problem is the aortic root dilatation and aortic aneurysms and dissections. But the tall stature is important. And I show you an example from Indonesia, which may be Marfan syndrome. I was in Sulawesi, Tartu Raja, and I saw these rock graves. And in this family, we see that some of the dolls need extra space. And in these rock graves, the dolls that we see here, here's a little door to the grave. We see that they represent the people who have been buried here. So apparently some of the people in this family were extremely tall. That's why they made this extra space in the niches for the dolls. So maybe this is Marfan syndrome. And the special photos and the family made this possibly interesting for a publication. If we go into this village and we find a new mutation in the Marfan gene, fibrillin 1, it's very difficult to get it published because that's not new. There are so many different mutations in this gene. But if you add a photo of a rock grave like this and you say, oh, well, this is the family, these are probably ancestors that already had the disease, then a scientific journal may find it interesting to publish it. Unusual but interesting, you think? So before starting your project, and that's my way of thinking here, define UOPs, units of publication. So a UOP is the amount of questions, data, and answers that is minimally needed for a publication. And we use UOPs to structure the research. And we will give you some examples of that. And you assign, if you have a group of researchers, of course, that's only possible if you have more than one person in your research, you assign tasks and create a timeline. Here's an example of list of UOPs from my laboratory a few years ago. And here we have the different subjects. Many are still organized around the osteogenesis imperfecta osteoporosis. Here we have Marfan syndrome. And we have the subject of the research, the priority of the subject, work to do, this is already finished, and then who will do this work? And here we put the initials of possible authors. And that's important because if you have a collaboration, which I noticed is very difficult in Indonesia sometimes. If you have collaboration between two universities, it's complicated because everybody wants to be first. And my experience with international collaboration is that it can be complicated, but if you think about publications in advance and you have a research project and you define some units of publication, you can already discuss authorship and sharing of authorships and dividing the number of publication and first authorship among the group. That's a very important issue to prevent problems, quarrels about that later. So think in advance. And also we think about what type of journal we look here that we look at here. This one he managed to get published in New England Journal, one of the highest ranking journals in the world, far above Nature and Science and Lancet. And that was because we had, I will show you later, this was a very unique study. So what is the message of the proposed paper? And what is the message? What will be the message of your proposed research? What are the questions that need an answer? What is the hypothesis I want to test? So this is an issue, the hypothesis in science, and it's the way of thinking of actually since the 1950s. Before that, science usually was descriptive. You did experiments and you described what you found. Nowadays, we work more like we formulate a hypothesis. We design experiments to check whether or not this hypothesis is likely. And that's a different way of thinking, but still, nowadays, it's still possible to do descriptive research so that you don't have hypothesis, you just describe your findings. And that's, for example, I work sometimes with the Hero Foundation on Bali. That's a new maritime research project. And if we find a new species in the coral reefs, and you're not having the hypothesis, this may be a new species. You just describe what you find. And that's the same when you have a new disease in humans, for example. You describe what you find, the phenotype, the symptoms. But more often nowadays, we have a hypothesis. And that's the question then in the research. If the results are not in agreement with your hypothesis, what are things? If you do a lot of research, spend a lot of money and time on it, but you get results that cannot be published, that is a loss of time and of effort and of money. And sometimes it is important to get the negative results published it can be very interesting even, because you need a new hypothesis then. And then this important question, consult a statistician, because if you have too few patients in your research, you cannot make significant conclusions. So the power calculation is very important before you get started. And too many patients will cost the necessary time and money. So there's a balance there, and you need a statistician, or you need to be very good at statistics yourself. So, a manuscript, very important, determine the field of interest. I just told you already the studies we did on the new gene in the little village. Genetics was important there, but you can also have studies where internal medicine is more important, etc. So you check journals in the field of interest, and you select journals based on the impact factor or the quartile and the expected importance of your paper. And read some papers from them. That will give you an idea of the structure of the research papers, an idea of the kind of language they use. And some journals have a very specific requirement of the style. And when we publish in the New England Journal of Medicine, for example, the editors often just call me on the phone just to discuss. We want to change this sentence in your manuscript because it doesn't. It's not in agreement with our style, but I want to be sure that it still means what you were trying to say. And that's very important for these journals. And then the journal's field of interest. This is general medicine. This is a journal of bone disease, genetics. So that's you have to see what field of interest is important for this kind of research. Well, the next subject is scientific writing. I will give you some examples of very common errors that are made by people who are not native in English and who are native speakers of Bahasa Indonesia. So there are web-based courses for scientific writing, for example, Stanford Online, but these are aimed at native English speakers. So the content of a course like that is aimed at structure of your manuscript, style of your manuscript. That's a much higher level. So scientific language is important. I will show you some examples of grammatical errors, use of verbs, use of verb conjugation, use of singular, plural, use of articles, the result, so the is an article, and the structure of your publication. And be short. When I have contact with scientists or students, and they have the first manuscripts from Indonesian scientists, often my first proposal is to remove about half of the text from the manuscripts. They use so many words. Scientific language is important. To read articles from scientific journals, don't use a lot of, instead use many. There are many things that we only use when we speak. In the spoken language, in the written language, you don't use a lot of. Be careful with the word of. I'll come back to that, but it's probably something to do with the word untuk in Indonesian, which can be translated into 5 different words in English, depending on the sentence. Don't start a sentence with because. I'm just showing some common errors. Very, very common in Indonesian manuscripts I see is consist of, and when you mean it consists of. Very common. Grammar is important because Bahasa Indonesia hardly has any grammar. And if you listen to the BBC or CNN and you can use English subtitles, you get used to the language. It will sound or look funny when you make an error. And when you hear and see the text at the same time, it will be very easy for you to remember that. The use of verbs. And there's a big difference between in Bahasa Indonesia and English and all other European languages, by the way. We have auxiliary verbs. I have walked. So this verb is saying something about this one. You are working. It has been described. I don't like this. So this kind of constructions, this is specific for English. I haven't seen it in any other language, but these other ones are very common in all other European languages. And the use of verb conjugation, also unknown in Indonesian. For example, the structure of DNA has been described by Watson and Crick. So this is the pluper, the past perfect. It's used when something has started in the past and also finished in the past. So if you refer to other papers, other research in your manuscript, this is the only way to refer to the findings of someone else. But for your own results, you use the past simple. We found the result was the mice showed that kind of constructions. So you have to think about it. This is a very important slide because it's such a common error that you have to use this construction. We don't have to use this when something has become common knowledge. So we have to use this that the double helix structure of DNA has been described or has been found by Watson and Crick because it's common knowledge. So we can talk about the structure of DNA or the double helix of DNA without referring to the original publications. Use of singular plural is important. Usually for plural, we add an S. So buku-buku is books. So we don't repeat words. Sometimes these plural are irregular. We have houses, add an S, but we have mice, and not mouses. We have women. And interestingly, in the written language, you have man. The plural is man with A, and the singular is man with A, the plural is man with E, but you pronounce it the same. For the word woman, the E here changes the pronunciation, so it's woman, women. And we have chicken, not chickens. But we do say chicks. So there's also an issue with singular plural, and that's a very common error, different verb conjugations. So if you have singular, he walks, he does, plural, it's they walk, they do. That's different. But also there we have irregular issues like here, he is, he was, and not he is or something like that. You going? So they are, plural, they were. So it's a completely different word, but it means the same. And then the order of words is different in English. And I use an example that's especially easy to remember for man. Wanita cantik is beautiful woman. So the order is, we don't say woman beautiful. And of course, you don't speak about beautiful man. That's why I use the example of an interesting man here. Or maybe we could say a handsome man, but that's different. But it's just for you to remember that the order of words is different. And then in more scientific language, the affected individuals is different. So here we start with individual and here we end with individual. Very important. But here is an example that I found in Rotterdam downstairs from my apartment in Jakarta. There's an exception here. We have baby potatoes free pesticide. What is wrong here? It's the order of words. Free pesticide in English literally is gratis pesticide. And it's a sort of hypercorrection here actually. So pesticide free, is a hyphen here, means no pesticide. So here is an example where the order of words is really important because it means something completely different. And they are grammatically, they are both possible. And then the use of articles. The results, specific is the, unspecific is a. So example, again, based on things you can remember, I met a beautiful girl yesterday. That's unspecific, but I have a date with the girl that I met. So that's the specific girl. And singular, plural, if we have non-specific, there's no article, no or the. And if it's specific, you can use the. So I met beautiful girls yesterday. There's not a or the. But I have dates, it's less likely, of course, but I have dates with the beautiful girls. So it's just to stay in the same example. Here is a specific group, so that's different. Then you use the. And in the scientific language, we used knockout mice, non-specific. However, the knockout mice showed, and then we can have the conclusion with the simple past here about our experiments with the knockout mice. Some other very common errors use the word of. When I read an Indonesian manuscript, of is nearly always used in the wrong place. It's hardly ever used in a place, but it belongs. That's why I said maybe it comes from untuk. I'm not sure, but also the word until is very often used in the wrong place. It's in English a word that can only be used in relation to time, until 5 o'clock. And not it ranged from 50 to 85 kilo, but from 58 to 85 kilo. That's not until. You need to remember this. And if possible, you should try to write in English and not translate from Bahasa Indonesia. It's difficult, you have to actually think in English then, but it's worthwhile to try that. Translating results in bad grammar and in 20 sentences sometimes. Structure of the article is important, and that's depends on the type of paper. So read journals in the field or fields of interest. Read, and that's a very important issue, follow the instructions to author. If you don't follow them to the letter, the editor will just discard your paper. This does not follow our instructions, so we don't need it. So there are examples of different types of publication, short communication, a research article, those are very extensive usually, case studies, a single patient or a single family, a review. So, and the structure depends on this kind of issues, this kind of types of paper. For a research paper, you usually have the construction where you have, the title, the abstract or summary, the introduction where you cite the literature, the methods, the results, discussion, and you compare your results here with the literature, but don't duplicate text from your introduction here, that's important. Acknowledgement is important, and then the references. And the references need to be compatible with the style of the journal. And you have to be short. This is an example, I mentioned it already. Watson and Crick, who describes the structure of DNA, one of the most high impact publications in the past century in our field. It's a single page. This is the most important paper in this field. in the past century, and it's only one page. And very interesting, there's a very nice understatement at the end of the paper. They do not brag, we have found the basis of genetics. No, it has not escaped our notice. But a specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. And another point I want to show you here is we have also been stimulated by knowledge of the general nature of the experimental results and ideas of Wilkins and Franklin. So Rosalind Franklin had done X-ray diffraction of the DNA, and from that you could calculate the distance between atoms, and that helped Watson and Crick to create their model. So nowadays it would not be accepted that someone with such an important contribution was just put in the acknowledgements and not be a co-author. Think of the message you want to convey. Can you put a conclusion in the title? For which audience is this interesting? Think of a target journal. This term I got actually from Prof. Sultana. Is it sexy enough? And that's also important for grant applications. So sexy means here, does it draw the attention? Would the title trigger you yourself to read the paper? So that the title of your paper is already important. How to select the right journal? No, this is just a repetition of earlier. Is it important in genetics, internal medicine, terminology, whatever? So a long time ago, I did a lot of research on Helicobacter pylori in the 80s of the past century, when it had just been rediscovered. And we managed to publish a lot of that research in a general journal, The Lancet, and it would not be possible anymore unless you find something very interesting. One of my PhD students at that time found that Helicobacter pylori had already been discovered 100 years before, in 1896. There was a publication from an Amsterdam doctor who wrote that if you had recurrent gastric ulcers, you had to take some gastric juice with an intranasal tube, and you would see spiral cord bacteria in the gastric juice. Has been ignored for 100 years. Read, look at impact factor, look at instructions for authors, check the style of journal, and write a very good cover letter. The cover letter is important, and I will give you an example. It is this letter to tell the editors why your manuscript is important. And it's something that people forget about, and they don't put a lot of effort in it, but it is very important. You have to be polite. You have to tell them why it's so important for the readers of the journal, and you have to convince the editor that it's worthwhile to take this in consideration. Here, this example is the cover letter for a letter to the editor that I wrote. I'm involved in many cases of suspected child abuse in Europe and the United States. There was a paper in the American Journal of Radiology that mentioned that osteogenesis imperfecta, the brittle bone disease, it's only used by parents it's so rare, it's not possible to see it in cases of suspected child abuse. It's only used by the parents to get away with abuse. And this made me very angry because I know a lot of these families and they are in trouble because of this stupid idea. So the part I made yellow here is very impolite, very aggressive. It's condescending and arrogant, I said. This is inappropriate for a serious subject like child abuse, and we certainly do not expect it in a journal with high quality standards like the American Journal of Radiology. So I took a risk here, but the effect was that 25 minutes later, after submitting this, I got a personal email from the editor-in-chief just asking me to change 2 sentences in my manuscript, and then it went into peer review, and the next day it was accepted. So being aggressive can sometimes be good, but it's not always It's not always effective, of course, but just as an example. It is 9 o'clock. I can stop here, or I can show you an example of published research.
I would think that another 5 minutes will be okay.
Okay.
Yeah. Yes.
I will just show this. And it's an example of published research from an Indonesian student in my laboratory. And this is Eva. She works in Surabaya now. And she lived on Bali. This is her husband, who is an orthopedic surgeon. And they met a family in a village near Ubud. For these people, this family lives in Ubud, actually. And these 3 guys are suffering of a very severe form of brittle bone disease, osteogenesis imperfecta. And Eva wanted to do an internship for her master's degree in my lab in Amsterdam. So she brought DNA from this family and from another family. Just a short introduction, osteogenesis imperfecta is a disease of the bones, often caused by defects in collagen type 1. We see it in 1 in 15,000 individuals. It involves a lot of clinical issues, but the most important one, bone fragility, sometimes blue sclerae. Sclerae are the whites of your eyes. But there are very severe cases. This baby had died, and it's lethal at birth. You see a lot of fractured ribs here, broken bones. This baby died at birth. And in the mild forms, you see the blue sclerae here, and you see often bad teeth also. It's also based on the fact that collagen is not very good. This is a patient from a family also from Bali who had also contractures of the large joints, and he could not stretch his elbows or legs very completely. Eva found mutations in a new gene, but bad luck, the gene had just been published by a group from the United States. However, this is the mutations that she found by sequencing DNA from the patients. And this is a software that we use. It's called Alamut for interpretation of results from DNA sequencing to see on the protein level what's happening and how conserved, for example, the sequence is in this field. And then she found a novel mutation in the family with Brooks syndrome, which is a form of osteogenesis imperfecta. But confirmation, the gene has been published as causative in a disease. It is still important to confirm that in a higher-ranking journal. So that's what we did. So we published in a high-ranking journal, and we realize now that there are many patients with no mutation in all the known OI genes. So there must be some genes that are missing. So there are families where we have over-modification on the protein level of collagen. Have these been misdiagnosed, or is this a new gene? So we have to do some. This opened also some more research. And at the moment, we're looking at some 40 families with osteogenesis imperfecta from Indonesia. Checked all the known dominant and recessive genes. And so far, we have 2 families. And we have no mutation, so we're whole genome sequencing. It's hopefully going to give us some new genes in this disease. Exome or genome sequencing. This is the publication of the results of Eva. So just as a student doing her master's thesis, she became first author of a paper in a high impact journal. Usually when there are a number of authors, the first one is the one who did most of the work, or often also the PhD student. The last one, that's me here, is usually the senior author who didn't do any of the work, who organized everything. So that's an important issue in the order of authors. And one more example, and this is a longer story, so I won't finish it completely. It's just I want to show that how it started. This is a dinner party. This is on the right here is Ferdi. He is an Indonesian student from Semarang, Undip. He did a master's at that time with us, later a PhD. And this is the group with which we were doing the research at that time. And next to me is Renate, and she was telling me at this dinner party, she was doing an internship. I'm getting a little bit bored. Can you teach me some new techniques? So I said, I will teach you an old technique first, which is we had a family referred for mild osteogenesis imperfecta with many affected persons, and which shows X-linked inheritance mode. So that was very unique. The males are affected, the females are affected at a higher age, and normally the females, so in between we have females that are not affected, and a man does not confer it to the next generation. So this is the paper that we eventually got published in the New England Journal of Medicine, and this is the old technique, linkage analysis. So we did segregation of DNA markers in the family. We found an area of the X chromosome that contained linkage, that contained the gene, and in that area there were hundreds of genes, so that's not really easy, but then we did, we checked the whole X chromosome, we sequenced the entire X chromosome in 3 affected individuals, and we found this lots of DNA variants, of course, but so one of these genes contained, and it was a very unlikely gene, actually, because it's expressed in nearly all the cells. It's very important in neurology, in the dendrites of our nerves, but it was actually proven that we could actually prove that this was the causative gene. This was very unique, but we needed an animal model, and there was already I will skip this a little bit here, but you can see how widely expressed this gene is in nearly all the tissues, and it's a protein involved in dendrites. So it's not really likely, but we checked 5 more families, and we checked 95 more families with male osteoporosis or mild osteogenesis imperfecta. We have so many because we are the only center in our country that does this kind of diagnostics. So we already tested thousands of patients. We found 4 families with pathogenic mutations in the same gene. Families, this is how severe it is. You can see that in the spine of a family affected man. You can hardly recognize the difference between the intervertebral discs and the vertebrae. We found mutations. We found loss of protein. We found difference in the structure of the actin bundles. And we think that it's important in osteocytes, in directing the direction, the position of collagen fibers in the bone. So loss of function of this gene will cause loss of structure in the bone and very weak bones. So we also checked, and then we needed an animal model, and that's what I'm showing you this quickly because there was already a group in Germany that had an animal model based on neurological diseases. I contacted them for collaboration. So that's why our final manuscript had a lot of co-authors, but that's because we needed many people for the collaboration. But eventually we got it published in the New England Journal of Medicine, and that was a very important achievement, actually. I'm proud of that, of course. This is the last slide. This is not that I love my students. We were asked to pose for a photograph for the website of our hospital in the Women's Heart Disease Awareness Week. So this is not a heart of love like in the Valentine. This is a heart for heart disease. That's it. I will try to find a button to stop sharing. I cannot.
All right.
Can you kick me out?
I think it has to be admin to stop sharing screen.
Yeah, but here I yeah, wait, yeah, I can't do better.
Yes, stop sharing. All right. Thank you very much, Prof. Gerald. Thank you very much.
Welcome.
Well, actually, you don't really use too much time because I remember we started 5 minutes late for your session. So you basically just use 6 minutes of the extra time, which is absolutely fine. All right. So yeah, thank you very much, Prof. Gerald. What a very, very interesting presentation. And to be honest, I'm also get a lot of knowledge and also refreshing on publication and research topic. And to sum up the presentations, I've made a few notes here that Prof. Gerald, I will speak in bahasa. Jadi dari presentasi Prof. Gerald tadi bisa kita konklusikan ada beberapa poin penting dalam linking antara riset dan publikasi, yaitu yang pertama kita harus start early. We have to start early. So kita harus memikirkan tentang publikasi, rencana publikasi sebelum kita mulai penelitian. Kemudian juga we have to ask ourselves, kita harus tanya pada diri kita sendiri, apakah ada curiosity, ada rasa ingin tahu yang akan kita mulai dengan penelitian kita. Kemudian apa relevansinya terhadap ilmu pengetahuan. Kemudian apakah topik ini layak publis gitu ya, dan juga yang paling penting yang kita perlu point out kan adalah what makes us special, apa yang membuat penelitian kita ini spesial. Kemudian tahapan selanjutnya adalah kita harus membaca ya, ikro, harus membaca artinya dengan mengecek literatur. Nah, di sini ada tawaran yang baik sekali dari Prof. Gerald that we can email him whenever we need to ask literature. I hope you don't regret your offers, Prof. Gerald. Because I'm pretty sure after this, there will be tons of emails coming to your inbox asking about literature.
Yes.
Yeah.
No problem.
Okay, so we witness that, okay. And then selanjutnya adalah sebelum kita mulai, kita teknikalnya kita harus mendefine unit of publication atau unit-unit publikasinya, dan di situ kita akan membuat rencana yang kita akan tuliskan dalam bentuk tugas-tugas yang akan dibagikan kepada tim-tim peneliti, dan juga harus mempunyai timeline yang jelas ya. Nah, selanjutnya untuk proses penulisan sendiri harus diingat poin-poin penting bahwa kita harus menggunakan scientific language. Kemudian harus hati-hati dengan grammar karena sebagian besar publikasi adalah bahasa Inggris, dan juga harus memperhatikan strukturnya. Dalam memperhatikan struktur penulisan ini harus melihat kepada target jurnal yang akan kita submit. Kemudian jangan lupa kalau itu juga harus pendek, tidak boleh kebanyakan. Dan yang paling penting adalah menemukan jurnal yang tepat untuk penelitian kita. Nah, selanjutnya yang harus kita perhatikan adalah kita harus dalam penulisan kita harus memperhatikan pesan apa yang ingin kita sampaikan, perhatikan target audiens kita apa, and I like the part when you say is it sexy enough, because mostly scientists are kind of boring, so when we say is it sexy enough, it kind of let us think out of the box, and juga we need to ask ourselves, apakah kita akan membaca artikel yang akan kita tulis itu? Would you read that article? So that kind of thought is very important. Okay, thank you very much, Prof. Gerald. So I think we gonna move forward to the next session. Tapi sebelum kita mulai sesi selanjutnya, saya ingin mengingatkan kepada seluruh peserta bahwa setelah selesai sesi kedua nanti akan ada sesi pertanyaan yang akan dibacakan, yang akan saya bacakan. Dan jangan lupa ada tawaran menarik dari Prodia, yaitu akan ada hadiah menarik pemeriksaan Prodia untuk 3 orang penanya. Saya dikasih bocoran bahwa itu ada tiga penanya terbaik, tapi saya juga dapat bocoran bahwa itu bisa jadi 3 penanya pertama. Jadi siapa yang duluan bertanya, ya? Baik. selanjutnya kita akan masuk ke sesi selanjutnya, yaitu pembicara kedua kita hari ini dari tim Prodia, yaitu Ibu Rina Triana M.Farm Apoteker, yang merupakan kepala penelitian research support head di Prodia, yang akan yang akan memberikan presentasi dengan topik Prodia in Research. Saya bacakan sedikit CV dari Ibu Rina Triana. Beliau menamatkan S1 Farmasi di Universitas Padjadjaran Bandung pada tahun 2007, kemudian melanjutkan profesi apoteker dan menyelesaikannya pada tahun 2008 di universitas yang sama. Dan di universitas yang sama juga beliau menyelesaikan Magister Biokimia Klinik di Universitas Padjadjaran, dan sekarang beliau sedang dalam program doktor di Universitas Padjadjaran. Untuk riwayat pekerjaan, saat ini beliau merupakan kepala research support di PT Prodia, dan juga menjadi reviewers di beberapa jurnal yang bisa kita lihat di Indonesian Biomedical Journal, Molecular and Cellular Biomedical Sciences, dan juga Indonesian Journal of Clinical Pharmacy. Salah satu jurnal itu saya juga ingat saya pernah masukkan paper tapi ditolak, Bu Rina. Baik.
Rina juga ditolak kok, Dok.
Iya, jurnalnya susah masuknya.
Iya, Dok.
Baik, terima kasih. Selanjutnya saya persilakan kepada Ibu Rina untuk membacakan presentasinya.
Baik, terima kasih banyak Dr. Nanda untuk perkenalannya. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang. Yang saya hormati Dr. Dedi dan juga Wakil Dekan, kemudian Dr. Musin dan para dokter yang telah hadir pada webinar kali ini. Saya izin share screen. Sebentar, Dok. Sudah ya, Dok ya? Sudah terlihat ya, Dok? Ya, baik. Terima kasih, Dok. Pada kesempatan kali ini saya mungkin akan cepat, saya ingin merefresh kembali, men-sharingkan kembali terkait dengan layanan penelitian yang disediakan oleh Prodia, di mana semua alat, semua metode yang dapat kami kerjakan, semuanya itu dapat dimanfaatkan oleh Bapak Ibu dokter yang mungkin sedang mengambil spesialisasi penelitiannya, kemudian yang sedang mengambil publikasi, dan yang lainnya gitu ya, yang sedang hibah untuk penelitian dan sebagainya. Nah, berbicara tentang penelitian pastinya tidak lepas dengan berbicara tentang biomarker yang sudah pasti diketahui oleh semua Bapak Ibu dokter. Namun sedikit mengulang sedikit bahwa biomarker adalah suatu penanda biologis yang dapat diukur dari pemeriksaan laboratorium yang menggambarkan suatu penyakit, di mana biomarker tersebut dapat merefleksikan kondisi tubuh baik secara fisiologis, farmakologis maupun proses aktivitas dari penyakit. Sehingga dan mengetahui biomarker itu tentunya dapat membantu untuk menjelaskan proses-proses yang ada di dalam tubuh. Kemudian mengapa
belum di slide show, Mbak?
Slide-nya masih slide pertama, Ibu Rina.
Baik,
belum
Dok,
di slide
sebentar,
show
Dok.
ya. Ya.
Ini. Baik. Saya lanjutkan kembali. Ya. Mengapa gitu meneliti biomarker dan juga melihat hubungannya dengan penyakit itu menjadi sangat menarik, karena seperti yang kita ketahui bahwa tubuh kita itu terdiri dari banyak sekali molekul. Ada 100 triliun jumlah sel, kemudian ada 6 miliar DNA base pair, ada 30.000 gen yang mengkode protein, kemudian ada 10 juta protein yang dalam tubuh kita, kemudian ada 2.000 protein yang berfungsi masing-masing itu berbeda dalam setiap sel, ada 60.000 reaksi per sel per menit, kemudian ada 100.000 event-event molekular, dan ada 50 atau lebih organ dan juga sistem organ. kita gabungkan semuanya itu membentuk suatu ikatan yang sangat kompleks seperti peta gitu ya. Dan suatu penyakit ini bisa hadir sebagai hasil dari kompleks reaksi dari berbagai macam adanya reaksi dalam tubuh kita tadi. Sehingga kalau kita lihat ternyata ada banyak pola, ada banyak jalan menuju Roma gitu. Jadi ada banyak jalan. Di sini kita bisa melihat banyak peluang untuk menemukan suatu biomarker yang baru, khususnya untuk riset yang kita bisa hubungkan dari berbagai macam reaksi tersebut. Nah, saat ini big trends biomarker topik untuk research di bidang clinician adalah terkait dengan marker-marker yang ada di sirkulasi yang terkait dengan gen ya terutama. Kemudian sekarang juga lagi eranya metabolomik gitu ya, yang dimana gut mikrobiota usus kita yang Human Microbiome Project itu baru selesai di tahun 2013 dan ini menjadi sangat menarik untuk diteliti dan dihubungkan dengan metabolit yang dihasilkan. Kemudian terkait vitamin D juga sedang hype kembali apalagi di masa COVID yang ternyata diketahui bahwa vitamin D itu memiliki berbagai macam fungsi. Kemudian nutrition marker yang banyak yang dikaitkan dengan multiple nutrition, kemudian multiplex testing di mana dalam sekali kita tes itu bisa menghasilkan banyak sekali metabolit dan juga terkait dengan menemukan cut off atau nilai-nilai rujukan untuk menunjang diagnosis. Dan apa sih layanan terkait dengan research yang ada di Prodia? Nah, untuk pengalaman Prodia sendiri di research ini kami telah mengerjakan atau membantu penelitian dokter gitu sejak tahun 1991. Jadi sudah lebih dari 31 tahun kami membantu peneliti dalam tes-tes riset, kemudian informasi terkait tes, kemudian metode dan juga literatur. Dan kami telah mengerjakan lebih dari 4.200 sejak tahun 1991 untuk tujuan-tujuan akademis, kemudian untuk studi epidemiologi dan juga untuk scientific publication. Kemudian kami juga berkolaborasi dengan 45 institusi di bidang pendidikan dan juga penelitian dan kami sangat berbangga hati gitu ya dengan dapat berkolaborasi dengan FKUSK hingga saat ini dan terus terjalin kolaborasi yang baik. Kemudian kami juga telah mengerjakan lebih dari jenis tes riset baru dengan berbagai macam variasi teknologi, metode dan selalu berkomitmen untuk menyediakan kualitas yang terbaik. Dan karena Prodia ini tersebar di seluruh Indonesia dari Sabang sampai Merauke di 259 outlet di 74 kota dan di 34 provinsi sehingga Prodia menjadi partner yang tepat apabila penelitian Bapak Ibu dokter ini research-nya ini multi-site gitu ya. Jadi karena Prodia ada di Sabang sampai Merauke ini dapat dijadikan rekan yang tepat untuk membantu penelitian yang dilakukan lebih dari satu site. Kemudian apa saja layanan yang disediakan? Yang pertama adalah kami menyediakan informasi tes riset. Jadi Bapak Ibu dokter yang mungkin telah bimbingan gitu dengan promotor telah menyusun usulan penelitian gitu ya. Tentunya perlu melihat apakah visible untuk dilakukan gitu untuk apalagi marker-marker yang baru gitu ya. Nah, ini kita bisa berdiskusi gitu apakah biomarker ini yang memang misalnya belum disediakan secara rutin di lab itu bisa dikerjakan gitu ya. Nah, itu kita akan explore terkait dengan itu kami bisa saja meminta literatur acuan dari para dokter dan nanti kami akan coba explore gitu ya apakah bisa diimport atau tidak terkait dengan reagen yang dibutuhkan. Kemudian kami juga memprovide scientific literatur misalnya Bapak Ibu menemukan jurnal-jurnal yang berbayar yang atau yang belum bisa dibuka itu juga kami bisa membukakan ataupun nanti kami juga bisa meminta kepada Prof. Gerard yang punya akses lebih banyak gitu. Kemudian kami juga memberikan informasi terkait penanganan sampel, metode, dan juga pemilihan teknologi. Dan ini juga penting ya, terutama terkait dengan etik ya. Berapa banyak nih darah yang diambil, kemudian spesimen biologi lain, urin, feses yang diambil. Dan ini juga bisa kami sediakan, bisa kami hitungkan Misalnya untuk pemeriksaan screeningnya, pemeriksaan tes risetnya, parameter ujinya segini, itu berapa banyak yang akan diambil. Kemudian kami juga memfasilitasi pemeriksaan menggunakan research use only dan juga lab development test reagent. Jadi bukan hanya yang in vitro diagnostik yang sudah rutin kami kerjakan, tapi untuk tes-tes khusus yang memang belum IVD gitu ya, atau masih butuh pengembangan lab development test, itu juga kami bisa fasilitasi. Kemudian kami juga membantu terkait pengumpulan spesimen dan juga penyimpanan untuk sampel research gitu. Jadi apabila sampel research kita membeli kit gitu ya, dan itu waktu dibuka harus dikerjakan nih di satu kit yang sama gitu. Karena nggak bisa kayak pemeriksaan yang sudah diterima untuk umum. Tadi dia harus dikumpulin dulu, dan nanti untuk menghemat dikerjakan pada saat yang bersamaan. Nah, itu kami menyediakan wadahnya untuk mengumpulkan sampel dan untuk penyimpanan untuk stabilitas yang lebih panjang. Kemudian kami juga untuk beberapa kasus tertentu membantu untuk analisis statistiknya, tapi memang masih terbatas karena orang yang mengerjakan ini hanya satu ya, hanya saya doang gitu. Nah, ini masih terbatas. Kemudian untuk publikasi sendiri, Prodia ada di bawah Prodia Education Research Institute atau PERI itu mempunyai sarana untuk memfasilitasi publikasi, yaitu INABJ ya, atau Indonesian Biomedical Journal. Kemudian untuk research yang Bapak Ibu mungkin sudah siapkan gitu ya, apabila memang di kami itu sudah termasuk tes rutin atau tes yang disediakan untuk umum, untuk pasien umum, namun memang masih mempunyai kebaruan yang tinggi gitu, itu kami akan memberikan keringanan biaya khusus untuk penelitian. Namun jika tesnya itu adalah tes riset yang di mana memang belum rutin Prodia kerjakan untuk melayani pasien umum, itu dokter hanya akan membayar biaya reagen, kemudian biaya bahan pembantu atau consumables yang memang dibeli untuk penelitian ini, dan juga biaya transportasi, biaya tube-nya gitu. Sedangkan untuk biaya jasa packing, kemudian jasa pengerjaan, SDM pengerjaan yang mengerjakan tes riset tersebut, kemudian biaya lembur SDM, biaya listrik, termasuk biaya alat dan maintenance alat itu tidak akan dibebankan oleh Prodia, tapi menjadi beban Prodia karena memang Prodia ini ingin memajukan penelitian di Indonesia. Jadi memang yang dikeluarkan untuk tes riset itu hanya biaya reagen dan consumables-nya dan juga penanganan sampelnya memang dikeluarkan untuk riset tersebut. Dan di Prodia sendiri kami mempunyai 7 lab, 1 lab rujukan dan 6 specialty lab yang semuanya dapat dimanfaatkan oleh Bapak Ibu dokter dalam melakukan penelitian. Yang pertama adalah lab rujukan nasional atau PNRL lab, ini yang ada di Jakarta. Di sini dia mengerjakan tes yang sudah rutin tapi yang sophisticated gitu. Jadi alatnya itu memang yang hanya tersedia di Jakarta saja dan yang memang masih memiliki kebaruan yang sangat tinggi gitu. Dan di sini kita di lab ini beroperasi 24 jam gitu, dan menggunakan otomasi jadi dari satu work cell gitu itu dapat terhubung dengan berbagai macam alat sehingga semua dilakukan dari mulai pre-analytical sampai post analytical itu semuanya terotomasi gitu ya, sehingga mengurangi human error. Kemudian yang pertama yang kami miliki adalah di flow cytometry. Di sini di lab ini ada alat flow cytometry, di lab imunologi ada alat flow cytometry yang dapat digunakan untuk penelitian, apalagi penelitian Bapak Ibu dokter terutama terkait dengan sel. Kami punya 3 alat dari BD Vexcanto, kemudian Vexvia, dan yang terakhir yang kami beli adalah Vexlyrics yang mempunyai pewarnaan yang lebih banyak lagi dibandingkan Vexcanto. Aplikasinya apa saja? Ini tentunya untuk melihat fenotipe di permukaan sel, kemudian produksi sitokin yang ada di dalam sel, kemudian untuk proliferasi sel, siklus sel dan apoptosis sel, dan yang terkait dengan sel gitu. Dan di COVID sendiri kami banyak sekali mengerjakan penelitian yang berbasis dengan untuk melihat memory T cell dan naif T cell dan juga Treg cell. Dan di alat ini, di flow cytometry sendiri, kami juga menemukan ada banyak kit yang research use only yang ternyata bisa dijadikan panel seperti panel imunologi ini, metabolik, kemudian ada panel human metabolic yang isinya itu kalau nggak salah ada sekitar belasan sitokain gitu. Kemudian ada kidney function panel. Jadi dalam satu kali running tuh kita bisa dapat banyak banget target. Kemudian ada immune checkpoint panel. Dan untuk rutin sendiri ini kami telah mengerjakan leukemia phenotyping dan juga HLA typing gitu, dan ini sudah rutin kami kerjakan dan bisa kami terima juga untuk penelitian. Kemudian untuk di lab specialty lab yang kedua adalah di lab research dan esoterik. Di lab research dan esoterik ini kami punya alat ELISA reader yang memang ini aplikasinya banyak sekali untuk penelitian karena digunakan untuk analisis sitokin tunggal, terutama untuk penelitian-penelitian spesialisasi atau S2 biomedik gitu ya, yang memang marker-marker yang digunakan nggak banyak-banyak gitu ya, biar cepat lulus juga, jadi nggak banyak-banyak juga markernya gitu ya. Jadi untuk analisis sitokin tunggal hanya satu yang di marker-marker yang digunakan ini bisa menggunakan ELISA reader, dan di Prodia sendiri kami punya 2 reader dari Bioret dan juga dari EasyRead. Kemudian selain itu di lab ini juga punya alat Luminex atau MacPix yang bisa untuk mendeteksi sitokain yang banyak gitu. Dia punya beberapa panel, tapi selain itu uniknya di Luminex ini kita juga bisa mengcustom gitu. Jadi misalnya di sini kita bisa lihat ada panel sitokain. Di sini panel sitokainnya banyak sekali, tapi di penelitian kita itu hanya butuh misalnya IL4, IL6, IL10 dan TNF alpha. Misalnya kita hanya butuh 4 dan ini kita bisa custom gitu. Dan dikerjakan di alat Luminex gitu. Kemudian untuk specialty lab yang ketiga adalah di lab molekular diagnostik. Di sini kami juga punya alat yang cukup lengkap di mana kita bisa lihat gitu ya, dari kita misalnya diskusi dulu berapa target yang para dokter ingin teliti. Misalnya targetnya hanya kurang dari 5 target gen, itu kita bisa menggunakan PCR RFLP, real time PCR, multiplex PCR dan yang lainnya. Kemudian apabila targetnya itu banyak, itu lebih dari 20 target, itu kita bisa menyarankan dengan microarray, dengan mass array ataupun dengan NGS. Nah, ini adalah beberapa metode dan juga beberapa tes yang telah kami kerjakan untuk penelitian, dan saat ini kami juga punya alat NGS ya, yang Nextseq dan juga MISEQ gitu untuk digunakan untuk penelitian. Kemudian specialty lab yang keempat adalah lab cytogenetik. Lab ini juga tergolong baru. Ini kami gunakan untuk dengan menggunakan metode karyotyping untuk menganalisis kromosom. Nah, ini biasanya Prof. Gerard nih seringnya di sini ya, di lab molekuler dan juga di lab genetik untuk penelitian. Kami juga punya specialty lab untuk lab patologi anatomi. Di lab patologi anatomi ini juga kami punya alat yang lengkap dan sudah diresmikan juga oleh Kemenkes. Di sini kita punya dari mulai pemrosesan jaringan, kemudian untuk pembuatan parafin blok, kemudian untuk mikrotom, potong-potong blok gitu, potong-potong jaringan, dan juga mikroskop untuk melihat jaringan, dan juga alat otomasi ya yang kita gunakan. Jadi bisa langsung menggunakan sel yang antibodinya langsung diotomasi gitu. Nah, ini juga mulai dari tahun kemarin kami mulai menerima penelitian-penelitian yang berbasis IHK gitu. Jadi kalau tahun-tahun sebelumnya itu belum pernah gitu ya ada yang penelitian IHK, dan kami sangat senang gitu karena mulai tahun kemarin ternyata ada juga nih yang tertarik di penelitian IHK gitu. Jadi ini mulai lab ini juga yang tadinya hanya untuk lab penelitian karena ada permintaan dari klinisi untuk membantu penelitiannya di IHK. Kemudian specialty lab yang terakhir yang keenam adalah lab MS, di mana lab MS di Prodia ini merupakan salah satu keunggulan lab Prodia karena berbeda dari lab-lab lain. Kami mempunyai lab MS yang cukup lengkap ya, seperti ICP-MS, HPLC, UV dan fluorescence, LC-MS-MS, dan GC-MS. Dan berhubung sekarang juga sedang maraknya gitu metabolit yang diperiksa, dan ini memang menjadi suatu alat yang lebih unggul gitu ya karena lebih sensitif dan juga lebih spesifik gitu untuk metabolit ya. ICP-MS biasanya untuk trace elemen, kemudian HPLC itu untuk banyak vitamin, MDA gitu ya untuk antioksidan, kemudian LCMS/MS untuk banyak hormon, dan GCMS untuk senyawa-senyawa yang mudah menguap atau volatil. Kemudian kami juga menyediakan bagi Bapak Ibu mungkin yang memulai penelitian yang bingung gitu, kita punya ada 15 brosur tren biomarker penelitian di masing-masing bidang, dari bidang internis, kemudian bidang kardiologis dan yang lainnya, dan ini semuanya sangat bisa didapatkan gitu, siapa tahu untuk bisa bahan bacaan gitu ya terkait tren-tren biomarker apa sih yang untuk terkait penyakit tertentu, ini bisa didapatkan dari perwakilan marketing kami di wilayah Bapak Ibu masing-masing. Kemudian ini adalah Indonesian Biomedical Journal yang difasilitasi oleh Prodia di bawah PERI atau Prodia Education Research Institute, di mana di INABJ ini udah Scopus Index dan juga publish-nya itu cukup banyak gitu ya, 4 kali dalam setahun gitu. Dan untuk biayanya sendiri sangat murah gitu dibandingkan dengan jurnal yang lain. Untuk submission fee-nya itu 500.000 dan publication fee-nya itu 2 juta gitu. Dan ini memang masih Q4 yang Scopus Index, tapi terakhir saya lihat di scopus.com itu sudah 24% ya, dan nanti mungkin di Juni atau Mei katanya sedang ada sedang ada review kembali, mudah-mudahan di Juni itu kita bisa di 25%, jadi bisa naik persentilnya menjadi Q3. Kemudian aplikasinya gitu ya yang saat ini di Prodia itu yang paling banyak gitu ya, ya tentunya terkait dengan COVID gitu, di 2 tahun kemarin banyak sekali penelitian yang terkait dengan COVID gitu. Kemudian yang menarik itu juga banyak yang terkait dengan depresi gitu ya, karena memang ditemukan banyak sekali orang-orang yang depresi gitu terkait dengan COVID. Kemudian terkait dengan penyakit autoimun, dan yang paling banyak itu juga terkait dengan penyakit diabetes mellitus tipe 2 yang memang prevalensinya itu sangat besar di Indonesia ya, termasuk studi S3 saya ini juga saya meneliti terkait dengan DM tipe 2. Nah, ini adalah contoh-contoh dari isi brosur yang tadi telah saya paparkan gitu ya. Nah, ini Bapak Ibu dokter bisa membaca apabila ada yang misalnya lagi mau tugas akhir gitu, ini bisa dibaca-baca, siapa tahu ada menambah pengetahuan gitu ya, biomarker yang terkait dengan penyakit tertentu. Nah, ini yang terkait dengan jantung, kemudian ini yang terkait dengan autoimun, kemudian ini yang terkait dengan pernafasan, ini tentang ginjal, kemudian tentang antioksidan, tentang lambung, dan semuanya bisa didapatkan untuk brosur-brosurnya yang terbaru kami mengeluarkan tahun kemarin itu terkait dengan metabolisme. Itu semua dapat didapatkan dari perwakilan marketing kami. Demikian yang dapat saya sampaikan. Apabila ada yang ingin menanyakan terkait penelitiannya dapat menghubungi research.support@prodia.co.id atau dapat langsung juga menghubungi Mbak Nirmala atau Mbak Emil di Prodia Banda Aceh, nanti akan diteruskan kepada kami di pusat. Terima kasih Dok, saya kembalikan kepada dokter.
Baik, terima kasih pada dua pembicara kita. Tadi Ibu Rina menarik sekali presentasinya ya. Jadi kami bisa memahami lebih jauh tentang support yang Prodia berikan bagi research ya. Bukan, saya lihat di sini bukan hanya untuk bidang medis, tapi bisa juga untuk berbagai macam bidang lainnya ya, yang bisa membantu untuk meningkatkan kesehatan masyarakat pada umumnya, dan juga pada khususnya itu untuk membantu meningkatkan profil penelitian di Indonesia ya, bagi para dokter-dokter di Indonesia, bagi para tenaga kesehatan, dan juga bagi pada semua pihak yang membutuhkan. Nah, karena kita sudah masuk ke tahapan yang terakhir, yaitu kita masuk ke dalam sesi diskusi. Ini saya melihat ada 3 pertanyaan dan ada 1 yang mengajukan tangan gitu. Tapi karena tadi yang mengajukan tangannya sudah dari tadi, apa mungkin boleh saya panggil dulu yang mengajukan tangannya? Dokter Maina Afrida, ini admin apa boleh bertanya langsung atau harus saya bacakan kalau ada yang ingin bertanya langsung?
Boleh saja,
Karena
Dok.
Boleh ya, jadi nanti bisa tolong di-unmute Dr. Maina Afrida. Apakah Dr. Maina Afrida ingin menanyakan, memberikan pertanyaannya langsung kepada dua pembicara kita? Halo, Dr. Maina Afrida, sudah di-unmute belum ya? Sepertinya tidak ada respon. Baik, tidak apa. Selanjutnya saya akan membacakan pertanyaan yang pertama dari Dr. Juwita Ridwan. Ini pertanyaannya ditujukan kepada Profesor Gerald. So the question is related to Brach syndrome. There's a mutation which is deletion 5p on FKBP10 gene. The question is with this type of deletion, what is the impact and what kind of biological process which is affected by this type of mutation?
Oops.
Is it?
Well, it's a loss of function mutation. So
It's loss mutation. Yeah.
Yes, so it's a loss of function mutation, and the gene or the protein is involved in processing of collagen. So it's a specific part of the processing of the collagen that's involved in making collagen ready for deposition in the skin or in the bone. And it's a very complicated process. It's hard to talk about it without showing slides, which I don't have available at the moment. But there are a large number of enzymes that are involved in processing of collagen. And I showed you this one picture of a bone with all the lines. That's collagen is important, like in a building, the steel fibers that make it elastic and strong. So without that, even a minor earthquake would destroy the apartment building. So that's why the genes for the enzymes involved in collagen processing are involved in recessive forms of osteogenesis imperfecta. So an FKBP10 is just one example. The Bruch syndrome is just actually osteogenesis imperfecta with the additional syndromic feature of the contractures of the large joints. And I do not know actually why this gene specifically has an effect on the joints. That's no. Okay.
All right. So in overall, it actually affects the collagen synthesis and also the function of the collagen itself. Is that how?
Yes.
Yeah.
It's a chaperone and needs to be processed in the endoplasmic reticulum and then put to transport. So it's
Yes. So it's affecting the whole pathway of the collagen metabolism.
Yeah.
Itself. Yeah. All right. I hope it answered the question, Dr. Juwita. And we move on to the next question from Dr. Azaki Abu Bakar. I think I will just read this question. Good afternoon, Prof. Gerald and Dr. Nanda. As a chairperson, I would like to ask you about tips and tricks in choosing the suitable journal for submitting our papers. Thanks for your kind attention. Maybe Prof. Gerald already put briefly in the slide, but I believe Dr. Zaki would like to know the tips and tricks for choosing the right journal.
Yeah, I mentioned it already in my presentation, of course. There are several issues there. The subject of your research and the subject of the journal, but also the audience of the journal. So is it a more general audience, or are you aiming at a very specific subset of scientists or medical professionals? And then, as I mentioned, read some of the papers in a journal that you are interested in. And sometimes it's also possible just to send an email to the editors that you are writing a manuscript on this subject. Do you think it would be interesting for your journal? It's that you can simply ask, like you can also simply ask authors for a PDF copy of their manuscript if you have difficulty of accessing that through your library. So it's a lot easier nowadays to send emails and ask people questions, and sometimes you don't get an answer because people are very busy, but very often you will get a reply. People are honored that you are interested in them, you know. And
Yes,
that's
yes.
but also the reading of papers in the proposed journal is important, just to see what kind of subjects do they have. Just go. It may be difficult when you have no online access to those journals. That's a problem, of course. But and that's more difficult to get access to. I mean, I can help you getting some PDF files of publications, but if you do not have access to the journal itself, but you always have there's always public access to the content of the journals. The abstracts are, for example, in PubMed online. You can always see all the abstracts of the publications, and that's already an indication of a specific journal. What kind of papers do they accept? Do they publish? Yeah.
Yes.
Yeah.
Okay. Thank you very much, Prof. Gerard. And also, I also had experience that it always nice to ask the publication, what you call that, committee, if they have like special issue topics. Yeah, it's also very interesting. Yeah, because if we're aiming at that special topic, they might sometimes they give us a little bit more like time to extend the deadline of the submission.
Yes.
Yeah.
Sometimes you get online, you get emails asking you to publish something in their journal, and you have to be careful with that because very often these are open access journals that ask thousands of dollars for publication.
Yes. Yes, predator journals they call it. Yeah. All right. Yes, thank you very much, Prof. Gerard. Okay, we also have one more question from Dr. Mauliza. She's registered in pulmonary division, so she's doing specialty at University Shah Kuala. Actually, this question is for both of you. So the first question is there any like tips for feasible research according to PRODIA research methodology, the applicable research within PRODIA? Because I know you both are with PRODIA. So is there any tips so we can like tailor our research that is suitable for the methodology which is available in PRODIA? And also the second question is if regarding the English translation, is there any like program or software in PRODIA where they can upload their manuscript and that can help them to check the English proficiency? Yeah.
Well, to begin with the last question, maybe. If you use Google Translate, that's the worst kind of translation program that's on the internet available. Actually, Microsoft Word has a built-in function which also looks at grammar, and that's a very good option actually. So, and also there is a program called Grammarly. It's available online for free. I don't know, Rina, is there any special software at PRODIA available for this kind of things? I don't think so.
Gimana Ibu Rina, mungkin ada special service?
Tidak ada sih ya Dok ya, cuman kalau kami tuh biasanya ke Enago tapi berbayar sih ya Dok ya. Kalau misalnya itu ya dok ya untuk untuk lihat
Proofreader.
translatannya gitu ya, proofreader gitu kita ke Enago, tapi memang ya berbayar gitu dok. Cuman kalau misalnya masukin ke INABC, apabila dari editornya itu misalnya accepted, tapi masih ada yang harus diperbaiki, itu biasanya ada pendampingan jurnal dari setahu saya ya dok ya di Peri itu ada pendampingan jurnal di situ dia untuk memperbaiki sama sih seperti Enago, cuman itu ya memang khusus untuk yang mau dimasukin ke INABC aja gitu dok. Karena saya bukan orang INABC, jadi di situ saya tahunya ada pendampingan jurnalnya gitu dok. Cuman kalau yang pernah saya lakukan itu terkait dengan manuskrip biasanya ke Enago sih dok.
Baik. Yang terkait pertanyaan pertama tadi sepertinya sudah dijelaskan secara ringkas ya oleh Ibu Rina bagaimana agar penelitiannya bisa sesuai dengan metode penelitian yang ada Prodia. Mungkin bisa dijelaskan secara ringkas lagi Ibu Rina.
Ya, baik dok. Sebenarnya dari kami itu sangat open to discuss ya dok ya. Jadi misalnya dokter tentunya misalnya tertarik meneliti sesuatu dulu, tentunya dengan bimbingan dulu dengan promotor yang terkait apakah sudah oke gitu markernya, nanti baru misalnya bertanya gitu kepada kami. Nah, kami akan sesuaikan dok dengan alat yang kami punya gitu dok. Jadi kalau misalnya alatnya punya dan kami juga mungkin butuh waktu untuk menanyakan terkait dengan reagennya gitu. Kadang alatnya ada nih tapi reagennya nggak bisa diimport, itu juga kan nggak visible ya dok ya. Jadi itu kami butuh waktu untuk melihat misalnya alatnya sudah ada dok, tapi kami coba dulu ya dok karena belum pernah dikerjakan sebelumnya. Tapi kalau udah pernah dikerjakan kami bisa langsung ya bisa dok di Prodia. Tapi kalau belum pernah dikerjakan kami tanya dulu ya dok ke vendor yang memang sudah bekerja sama dengan kami apakah bisa diimport atau enggak. Tapi kalau misalnya bisa, itu bisa kami pasti infoin bisa gitu dok. Tapi kalau nggak bisa ya agak butuh waktu lama lagi, kami harus cari lagi nih vendor-vendor langsung ke reagennya dia punya nggak ya jalur khusus untuk import ke Indonesia. Itu sih dok yang biasa kami usahakan dok.
Ya, baik terima kasih. Jadi intinya Prodia selalu terbuka untuk diskusi. So you can help anyone that want to do research with Prodia. That's the conclusion from Ibu Rina explanation. Okay, I think we almost running out of time. I'm not sure if we can go over time. All right, so we can do 15 minutes max. So I think we should be okay. We still have. Well, this is
Okay with me.
This is. I was worried that suddenly we might get cut off.
Oh yeah,
from this online platform. That's the worst. What could happen? Okay, actually, for the last question, I have one burning question from myself, Prof. It's regarding in your presentation. You mentioned about the importance of having hypothesis, but also there's also possibility to do descriptive research where we just explain our result. For example, like what Watson and Crick done in the past when they describe their finding about DNA structure.
Yes.
So as it's moving forward in this kind of research area, how is the value of descriptive research itself compared to the hypothetical research on your point of view?
Well, I think the descriptive research can also be very important. For example, in the field of genetic diseases, we see that Indonesia has, I already mentioned, 354 recognized ethnicities that are more or less genetically isolated outside Jakarta and Semarang in the big cities. And that can have a huge impact on what you will see in genetic analyses. I have looked at some diseases, for example, cystic fibrosis, because I thought that the cystic fibrosis should be more common in Southeast Asia because the carriers are protected against dehydration in case of cholera or typhoid fever. And that's also has been common in Europe, but also in Southeast Asia. So I checked the genome aggregation database, and I saw that carriers in the database, there are no patients in them, carriers are present, and but the mutation spectrum between Europe and East Asia is completely different. There's no overlap,
Yeah.
except maybe in Indonesia because of the influence of some Dutch genes. Maybe we don't know that, but I'm going to describe that because it's important. Because until now, the common idea in publications is that there is no cystic fibrosis in Asia, in East Asia, and that's nonsense. So this is not based on a hypothesis, it's just based on looking at the pathogenic variants in the database and describe what's happening. And so I based on this, I expect that cystic fibrosis, mucus disease, is as common in Southeast Asian countries as it is in Europe. Only based on the fact that the mutation analysis has been done like 20, 30 years ago, based on East Asian people, based on the European and American mutation panel. It's completely different. That's descriptive research. So I think I'm going to do the same with hemoglobinopathies. Thalassemia is very common in Indonesia, but so far,
Yes, it is.
and I'm going to write a review on that. What we've seen that between different subpopulations, between different islands or between different provinces of Java, there's a huge difference in the causative mutations.
Yes, that's yeah.
And that's that kind of
that's very
descriptive
interesting.
research, especially in the field of genetic diseases, is very important.
Yes, that's very interesting. Thank you, Prof. Talking about thalassemia, I'm also the carrier of alpha thalassemia alpha trait.
Oh yeah,
I found
It's so
out
common.
it is so common. Yeah, just noticed it because we had the family gene check, so that's quite interesting.
Yeah.
Yeah. Okay. So selanjutnya tadi yang saya tanyakan adalah tentang bagaimana deskriptif analisis, karena memang kita di Indonesia lebih mengutamakan hypothetical research gitu ya. Tadi Prof Gerald sudah menjelaskan bahwa ternyata deskriptif research juga sangat penting untuk bisa dilakukan dan juga mempunyai value research yang sangat tinggi untuk menjelaskan fenomena-fenomena penyakit yang terjadi gitu. Bahkan yang sudah pernah diteliti sebelumnya juga masih bisa kita lakukan deskriptif analisis di tempat yang berbeda. Jadi mungkin bisa jadi ide juga buat teman-teman di sini untuk bisa memberikan idenya. Lalu juga saya akan mencoba menyelesaikan dalam 2 menit lagi. Untuk Ibu Rina, saya tertarik karena bidang saya adalah proteomik, Bu Rina ya. Saya bidangnya adalah di bidang I'm a proteomic scientist, Prof. So I'm working with mass spectrometry to do the proteomic analysis, shotgun proteomics. So pretty much the same like what you done next gen sequencing. But I haven't seen. Saya belum melihat that facility available in Prodia. So I'm looking forward to see that research field will be more expanding in Indonesia.
Oh yes,
yeah. It's very interesting because from what you explained about the gene mutation and so on, it will be nice to see what type of protein which is actually expressed or suppressed in one type of particular disease. Yeah. So hopefully that will expand. I think there's no more questions on the question and answer sheet I've seen. So now it's time for me to end this session, and I will return the session to the MC, Dr. Muhsin. Thank you very much, Prof. Gerald. Very nice to be in the same.
Nice to do it.
Yes, nice to be in the same online session with you, and also Ibu Triana. Thank you very much.
Terima kasih, Dr.
Oke,
Silakan, Dr. Muhsin.
Ya, silakan. Terima kasih banyak Dr. Nanda telah memimpin diskusi kita yang sangat menarik sekali menurut saya ya. We have a lot of new information, very nice presentation from Professor Gerard dan Ibu Rina Triana. Terima kasih saya ucapkan sekali lagi kepada pembicara dan moderator kita pada sesi ini, pada kali ini. Banyak ya disimpulkan oleh Dr. Nanda tadi dari presentasi dari Profesor Gerard dan juga dari Ibu Rina. Sekali lagi saya ucapkan terima kasih bagi moderator dan juga bagi pembicara atau panelis. Saya mengucapkan terima kasih juga kepada Dr. Dedi Syarija MKes dan Ibu Marisa Arifin MKes yang telah mendukung kelancaran kegiatan kita pada kali ini. Di Podia, mungkin banyak hal yang bisa kita lakukan pemeriksaan untuk apakah itu untuk medical check up maupun kalau misalnya kita ada ada gangguan-gangguan yang ingin kita periksakan secara laboratorium ke Podia ya. Untuk pemenang door prize saya akan bacakan ada 3 pemenang. Yang pertama adalah Dr. Juwita, Biomed spesialis neurologi, yang tadi telah memberikan pertanyaan yang sangat bagus sekali tentang beberapa tentang Book syndrome ya. Kemudian yang kedua adalah Dr. Azaki Abu Bakar, SPPD, resmi dalam konsultan gastroenterologi enterohepatologi. Dan yang ketiga adalah Dr. Mauliza. Selamat nih kepada ketiga pemenang yang nanti akan mendapatkan e-voucher pemeriksaan lab senilai Rp1.000.000 dari Podia. Nanti panitia akan menghubungi setiap pemenang door prize paling lambat 1 hari. Nanti mudah-mudahan bisa dimanfaatkan dengan sebaik-baiknya ini pemeriksaan pemeriksaan lab di Podia. Dengan demikian, dengan berakhirnya sesi diskusi kita dan pengumuman daripada pemenang yang mendapatkan door prize ini, maka akan berakhir pula lah kegiatan kita pada hari ini. Sekali lagi saya selaku MC pada kegiatan kali ini mohon maaf jika ada salah-salah kata dalam memimpin ketika awal-awal tadi. Kemudian saya serahkan kepada moderator. Kemudian saya juga mengucapkan terima kasih kepada semua pihak yang telah membantu kesuksesan kegiatan ini.